Great, thank you. I'm going to go ahead and have you invite all the speakers to join up here for the panel discussion, and also remind all of you here in the room to please go to the microphones when you're asking a question. And for the folks who are joining us through Slido, one great way to draw more attention to a particular question, if you want us to ask it here in the room, is to like that question. That moves it up in the line. So we'll be relying on the discussion here in the room, but also be looking at those questions to ask as well. And actually, to go ahead and kick off that discussion, I was going to go to a question that we had online that talked about sort of the last two speakers. Thank you. From the last two speakers, Vicki talked about a mechanistic model and relating that to the aggressiveness of breast cancer, and Amy mentioned the natural history model for CISNET. If you could talk about how these types of models are similar or different, and then also there was a related comment about how can these data and models be linked in the future. Is that possible?
So I think that these are really complementary models, and I think we need to really look at these complex issues from different perspectives. If we don't really know what the biology is, then we don't know what the intervention is. If we don't know the biology of what makes the mammogram work or not work, then we're going to just keep on doing mammograms in women who we are not going to get a benefit, and we'll get a skewed view that mammogram is less effective. So I think that this is a partnership hand in hand that really needs to occur, and I was just very excited to hear Amy's talk.
Thanks. So in Model W, we have a Gompertz-like function for the tumors to grow. At the beginning, when the model was being developed, the tumors would actually grow so fast they were the size of the moon too quickly. So we had to find a way to have an asymptote for tumor growth. So the bottom line is it's essentially unobservable. We don't just let tumors grow, and so those unobservable things are really where it would be great to be able to explore that more and to have biological data to drive those decisions.
We really don't know a lot about the temporal development of these biologically aggressive cancers, because typically a woman has normal imaging and then comes in with some, locally advanced breast cancer. We know a lot about cancers that develop slowly, but without the data to see what the drivers are and what happens first and what happens next, it becomes really difficult to model. But conversely, if you can't start modeling the data you just have a lot of case reports and no unifying theme.
I'm not on that topic, so I can't address it, but I did wonder, are there other ways to get, obviously if you have a woman who has cancer and you screen and you find it very quickly, you're going to have to act on that. You have a moral obligation, so you're not going to be able to watch it grow. Can you do an animal model or something else to try to get at those, now that you know some of what to...
Well, I agree ethically we don't let cancer sit, but what we have, which is, I didn't show a lot of the data, is we have women, particularly BRCA1 mutation carriers, and women who are Black African American women who will have small lesions, and we biopsy them. They are morphologically normal, and we got the right spot, so we're not doing anything unethical. And then we come back, and especially if we have like a couple of different lesions in line, because that's where it's very difficult. If you take something out, you can't say how does that influence what happens next. But if you have a vascular pedicle with 4 little spots on a string, and you take 2 of them out, you're sure you got them, they look normal, the person comes back, then you can really assign a temporal rate of progression, not to let the thing sit there after you find it, but to say how fast did it go from these little normal spots to, and that's for us, that's the thing that keeps me up at night is, pathologist says it's normal, I go tell the patient, and then they come back with cancer, and somehow I'm missing something.
So it's a question for Amy, and you can see that these are trending, the specific questions. And what probably jumped out at me and a lot of people was your response when people asked for your code, and that you were like, no, and that's showing up on this. But also, someone asked about how do you get other people to test it for reproducibility. But I guess I have a broader question, and that is, if something's NIH funded, we're sharing the data. Shouldn't at some level the code be made available?
Yeah, we struggle with this. So I don't program in C++. I don't know what it would be like to get something that's 20,000 lines of code and make sense of it, especially if it's not well annotated. So we certainly try to, again, it's like cleaning my desk. I should clean my desk, but the choice is, email, clean the desk, work on a paper. So yes, we would love to be able to have something that transparent and to be able to post it. We can't just give someone a password into our GitHub site. So I don't know if Dr. Foyer wants to make a comment about this, but we do many other things to try to make the model as transparent as possible, because that's been a problem. If it's just viewed as a black box and people don't know what's going on, then they're not going to trust the results. And so there's many things we try to do to make that understandable. And we have worked closely with some groups who have made models very closely aligned with ours, but to be a responsible partner in science, it would have to be a collaborative relationship and require our time. And I think people who have large data sets oftentimes struggle with that too, with privacy and identity. There's other issues, but it's not always responsible to just turn it over to someone and expect good things to happen because of it.
First of all, best talk I've ever heard on CISNET, so thank you, including all of mine. So it's probably good. So just to say this, we've been really struggling with this issue, and we want to do it responsibly. We had people who have gotten a version of our model and then started changing individual parameters, but then they don't realize that these parameters have to go together as a set. So we're actually, we have a programmers group within CISNET. We just probably don't know because we just set it up 2 days ago, but we're having a session on model accessibility that we're going to invite people within the community. So we try to do detailed documentation. We really try to, and people want to share code in the context of collaboration. And then we're struggling with the next step and how to do that step responsibly. So there's the issue of people not understanding all the subtleties of parameter sets that go together. The other thing we really struggle with is industry and the industry use of models, and just struggling with what's the motives of some of that modeling and what's the credibility of some of that modeling. And so we don't have easy answers for those questions, but I tell you, we're certainly struggling with them and want to push this forward as quickly as we can. There's things that are just pushing in different directions, and we're working on it. And great to have suggestions for that, because I think that's something that there's been a struggle with across the modeling community.
Amos, I just wanted to point out that Leah has been leading a group with Bo Peng that coordinates all the different simulation models that have been published and identifies what can be shared. And I think that kind of transparency is something that we need. I understand the concern about not releasing code, but longer term, it sort of limits your ability to have an impact to not be able to share. At least maybe you could share some modules, and that might also allow better sharing within CISNET if the modules can be swapped among the groups. So.
I think you're talking about genetic simulation resources, but we do try to share modules. For example, the lung group has had, and some of it's sometimes the inputs. The lung group has spent extensive amount of time doing something called a smoking history generator, And you put in a person's year of birth and their gender and it's going to even get into income and race, and it will simulate their lifetime smoking history, when they started, when they stopped, how many cigarettes per day on average they smoked. And that's a great resource for all kinds of modeling related to smoking. So we've been trying to do that when we can. And just hopefully the threshold for collaboration when we do share code is pretty low versus just somebody taking the code and running with it. So trying to get that threshold, I don't know if it's lower, so that if somebody comes to a CISNET group, sure, we'd love to share, but it's a little bit together so you could have all those subtle conversations.
David Conti, USC. Just an additional comment on the software, though, is, I think to raise that bar of sharing programs takes resources, both from NIH and from the investigators of writing a grant. Coming from the statistical world, we used to sort of create statistical models, write the paper, and the code was sort of an afterthought, right? Now, though, we need to make that part of our grants, part of our research programs, to actually create software that follows coding standards, can be released, can be used, so you have documentation that says, well, if you change this parameter, you need to understand that you're doing, and that takes time, and so it really needs to convert sort of our little research programs into software development type programs if we're really looking at how to sort of distribute what we're creating.
I had a separate but related question. Yamile Molina from UIC. I was just wondering, since CISNET's real focus is to evaluate interventions and the comparative effectiveness of them, what are current stances on incorporating increasingly evidence-based approaches and strategies to promote screening but also reduce diagnostic delays and information like that, if there are any, really in terms of, maybe specifically in terms of patient navigation? Thank you.
I'm not sure, you might have to repeat the question for me, but in terms of comparing which approaches to increasing screening, or?
Just in terms of increasingly standard interventions. So for example, if we want to be looking at multi-level interventions, right, how are perhaps the CISNET-affiliated teams working on multi-level intervention approaches that are becoming more popular? Patient navigation is one of the common folds in terms of breast cancer as well as colorectal cancer. So how are these increasingly important intervention strategies that are already being disseminated, how are they being incorporated into CISNET modeling?
Yeah, I don't know if we've done a lot of that type of thing. I know one area where we've started working on are these workflow issues. So just to give a very specific example, in terms of comparing batch reading of mammograms versus online, I guess is how it's referred to. So the process of how breast cancer detection and how to reduce these false alarms, and if we're really concerned about quality of life issues, looking at that. And so in terms of those multi-level, say, patient navigation, we haven't done that, but that's certainly something that we've written grant applications about and we're interested in.
Another question I wanted to ask, and Amy, you talked about measures matter, and this is something we heard Bruce talk about earlier also. And I just wondered, for all of you, what advice you might give research teams as they're thinking about refining their research questions or their frameworks to select the most appropriate measures and tools that appropriately reflect the intended construct. Or another way also to think about it is, are there new methods and tools that are needed for some of the questions that you're interested in? It's for all of you. I mean, I know, Vicki, you had talked about, new tools that you might be interested in, and Amy had talked about, sort of how the measures that are used and how those variables are constructed really matter. Mm-hmm.
I think that starting to look at issues of time and space are really critical for modeling and being able to model upstream and downstream. So at least from my perspective, the two tools that I outlined were ones that really could start to get at that question, where you could really dynamically take a woman who had areas of neovascularization, give them like a sugar bolus, see if you have increase in redox potential, increase in blood flow, and then you can go and take that area out as per standard of care, and you could really start to look at the biology of what this lesion is in the person, in and then out. And I think that, it may sound like really technical biology stuff, but I think if we're going to start to understand how obesity and pregnancy and insulin resistance and disparities in food deserts are all drivers of bad biology, I think having that dynamic and spatial information is really important.
So, yes, it may be hard to come by the data. And that's why one of the things that I showed was the longitudinal data sets, because where are you going to get longitudinal big data? That may be one of the kind of key pieces. Another, it may be a little bit far removed, but it's much more at the population level than the biological level. But there are people, Andrew Curtis, and I think he's at Kent State, looking at contextualization of spatial data. So looking at over time and space. So he has people walk through neighborhoods and capture what the context of that space is to give it a much more deep contextualization. So those are the kind of things I think we need more of. I don't know exactly what those would be in the biology realm, but I do agree that better incorporation of time and space. But you got to partly go with the data that you have, but you can use the models to help you figure out what you should be looking for next, because there's a temptation to put everything into the model and be over-inclusive and just try to get so close to reality that you're challenged by the same thing you started with, which is the complexity of reality. And so you've got to make decisions about how to make it simpler. Time dynamics can be extremely important, and some of the challenges include things like delays when you don't see something right away, or in your case, you're seeing it sooner than you expected. So then it's a knowledge about when to measure. But the models can help you figure out what those things are, because you can run the sensitivity analysis to determine how much of a difference it would make if you had that. And if it doesn't make that much of a difference, you may decide that that's not the thing you're going to go for.
Hi, I'd like to go to one of the online questions, and it's they're interested in your thoughts on how to nurture the pipeline for junior investigators interested in modeling and systems FE.
I'll say one thing is that someone mentioned data science. I think that's a great place to go. I think we need to make more places where the different disciplines can come together to learn from each other. So more specific training. I know Liz of Genexi is here. She's got a funding announcement out to do that, to develop T32 programs to teach in system science. I think great job, Liz. We need more of that. And places for people to kind of hang out with each other, make it a norm. So maybe having some sort of sharing platform, and where not only we share code and visualizations and data, but also we start to have conversations, kind of like a watering hole where people can get together and transfer their ideas. That's part of it too. And then there's also training for some of the end users. I think Bruce mentioned, he mention the UN? Someone, I think today, mentioned the UN as a consumer, as well as there'll be others, the journalistic community, the media, who wants to put it out there. We need to figure out ways to basically train them too, in a way, because if they're going to be consuming it, we don't want to have these same kind of problems come up where the models are misused. They're hard to understand, but we've got to figure out ways to convey the right amount of knowledge. And I guess I'll say one other thing. I think I've said this before, but it's about to some degree a comprehensive knowledge. So not just, I'm going to learn agent-based modeling. I think agent-based modeling is great. I love it. But I think that if you only learn that, you'll become more siloed. And then we also get these camps of what modeling approach is better, which is kind of ridiculous. It was the slide that Bruce had about winning. It's not about winning. It's about how do we get together and make the most out of that whole Method Man toolkit together. And so if you don't know what the toolkit is, all of it, at least some general knowledge, then you use your own hammer at your peril.
Yeah, I was just going to say in SysNet, I think what's worked really well is if someone comes in with a K award or their first R01 and they have funding, time for themselves, and maybe a little money to give the modelers, that has worked really well. We're very welcoming of that and able to work with them often in the, senior-junior relationship. But I think where we can still work on is trying to get the junior people together across the different organ groups and have a community for them. That I think more could be done to help their career development that way.
I just wanted to also note Liz Genexi is here in the room, and she posted a note with a link with some more information. So please see her if you're here, and also, for those of you who are on the WebEx, please follow that link and check that out.
So I think the point you just made about the junior investigators, I think that's super important, and we need more generally a community for our health modeling, community. I think it's great to have a meeting like this, but what's interesting to me is that I go to another meeting and I see completely different faces, and so it's sort of like we have these silos even within the modeling communities, and so that's, unfortunate. We have some people that run across them, like Bruce mentioned that he was in all the infectious disease modeling, work, but not very many of them are represented here, are they, other than Bruce? I mean, so we need, a couple maybe, but we need some more overlap. So what we need is maybe it's time to develop, and I've said this at the last place that I was giving this type of, having this type of conversation. There's a group at Pittsburgh that had a population health, I don't remember what it was called, but System Science and Population Health group meeting, and I raised the idea we need to have kind of our own association, our own meetings, our own journal, and not because we have to do something separate and different, but more like to try to bring everyone from all the different areas together. So I'm not trying to say we need our own thing, but more that we need a place where we can all commune and share and do it on a regular basis and in a systematic fashion and learn and hear from each other. Someone else mentioned today the Chris Backrack's group, the Interdisciplinary
Association of Population Health.
There you go. The Interdisciplinary Association of Population Health Science. And that's another group that needs to be, brought in with the other groups.
Bob Hyatt, USC. This may be a question for Amy, but others too. It was brought up in my mind by Vicki's presentation of using other sorts of data from imaging. But my question relates to animal model data. And so Amy's worked in the breast cancer and environment research program, as have I, and the idea of using animal models is to help us understand exposures that may be short-term and what kind of biologic effects they have that you can't study in humans. So I just wondered if there's been any work done in modeling groups to incorporate data from animal model studies into understanding human biology systems.
I don't know in the breast group if that's happened, but that would be a wonderful way to have the different arms of my research come together if that was the case. Maybe you have an example?
Yeah. I think the whole issue of pregnancy and pregnancy-associated breast cancer and pregnancy in the development of obesity and insulin resistance can be really looked at in a way in mouse models that are very difficult to do in human studies. I think we always have to have validation in human studies, but I agree with you. I think that, you can start to take mice who are obese and go through cycles of pregnancy. You can induce transgenes in the epithelial cells. You can look at how that affects it. And there are some really cool models right now called like the fat attack mouse, where you're able to, when you give a particular ligand, you're able to go and get rid of all the adipocytes in the body of a mouse. So these are things obviously you can't do in a human being. And I think that it really there's so much of a cultural and language gap between biologists, physicians, modelers, epidemiologists, that it really calls for people to learn each other's language and get together.
So my question is not that you can't do great things with animal models and learn from them, but the data that comes out of an animal model system needs to be integrated into some sort of mathematical language that works for human knowledge advancement as well. That's the question I think that's out there. It's in environmental health a lot. Rocky's getting ready to answer this.
So we had an announcement a while ago, it's now unfortunately expired, called Bridging the Gap Between Population Modeling and Biological Mechanism Modeling. And it was sort of a collaboration between us and the Division of Cancer Biology. And Marissa, I don't know if she's an example of an award winner for that in oropharyngeal cancer. Do you just want to say just a word, maybe step up to the microphone, just say a word about the type of thing that you've tried to integrate in that type of modeling?
That program, it was really fantastic in that it sort of, like you were talking about before, the need to have these different groups talk to one another and interact and do things that kind of cut across. So there were sort of two, the CISNET and then the sort of like more molecular scale modeling groups. This program was intended to kind of get that to kind of smoosh together a little bit, which was really great. And so what we did with that, we're looking at HPV and oropharyngeal cancer. And sorry, I'm using my lecture voice even though I'm on the mic. I'm sorry. Probably that's very loud. But anyway, even though so basically we're blending things from sort of taking it from the population level for HPV transmission dynamics down into the within host carcinogenesis process, and then back up to look at cancer incidence trends and really try and bridge all those things. And so we have data collection at all these different scales, including doing lab experiments and things like that in cell lines, as well as collecting data from participants and stuff. So anyway, that's my little small.
Thank you for talking about that. How did you get the different disciplines at the table? That's the challenge, right? That's one of the challenges.
Challenge. So it helped that the group that we started off with were already fairly interdisciplinary and a little bit used to the hilarious multi-jargon back and forthing between different disciplines. But I think that's really one of the big, everybody has talked about silos and the importance of bridging these things and trying to interconnect, and it's a major challenge. I don't have there's not a recipe, I guess, or at least I don't have one, but I would love to hear from people.
Well, some of the ingredients are, I think, time.
Totally.
You've got to get the people together for a period of time. They can't just, I tried many years ago, I hosted SBP, which is Social Computing, Behavioral Modeling, and Prediction. I was the chair for that in, I think, 2010. And I thought naively at that time, we really do need to get more of these content area experts together with these modeling experts. So let's do some very innovative thing where we do these speed dating roundtables, right? So you just bring them together and get 15 minutes at the table. They can all introduce themselves, exchange business cards, and move on, and we'll very deliberately mix them so that they're meeting each other. Well, they met each other, but nothing happened. And where it really happened was when I hosted Institute for System Science and Health, which was a week-long learning institute, which wasn't specifically intended to be an incubator, but it ended up being an incubator for a lot of these teams that developed because they were there for a whole week, kind of a captive audience for a week with the modelers and the content area experts that were trying to learn the modeling.
So hard to know what's going to be useful when you only have a short. Anyway, sorry. Keep
Yes. So part of it is time, but the other thing is people like Rocky. I have to say one of the things that was missing from your great slides was the facilitators. Is you don't know what you're missing when you don't have someone like Rocky, somebody who can think outside that box and be so successful at making it all come together and having it happen for this great long period of time. I hope you've gotten many NIH medals for that, Rocky, but you really do because, there's very few people who've been able to do this. Irene was one, and I think Grace is another one, Grace Peng, but these are the special people. We need to cultivate those, because if you don't have the people inside of NIH with the funds and the people special skills that it takes of being able to put those things together, then you can't do it alone.
I'm turning so red, I'm going to leave the room.
Rocky, do you have anything to add about how to pull together such great diverse teams and facilitate?
I'll just say one anecdote. So when a person from Division of Cancer Biology, which is pretty far from my expertise, and I got together to try and come up with this thing, we didn't understand. People from Division of Cancer Biology, we started talking. Jennifer Couch from Division of Cancer Biology, a wonderful person, and we started talking, and we couldn't understand each other, and people from Division of Cancer Biology says, what are you guys doing? You've been at lunch, 100 times, and so it took us probably a year of talking to come together enough to know how to make an announcement that would make sense. Mm-hmm. So that's just an example of when you go across those disciplines, how long it takes, and now we're pretty good friends.
Right. Well, in that spirit of taking time to talk across our disciplines and across silos, I'd like to encourage everyone to transition to our next event, which is lunch, and also to thank our speakers for excellent presentations and also discussion here. So thank you.